Next, we have Hilary from the board, not the board.
All right, thanks so much, Sam. This has been a super, can you hear me okay? Yeah. A super interesting conference already, and we're, not even at noon yet. So today, I wanted to start by talking about, in the context of genetic agency, we've got these two problems, right? Which genetic changes to make and how to make them. Though, I guess as we were speaking about earlier, then there's a lot of regulatory and so forth. And what I'm going to focus on for this talk is the question of which genetic changes to make. I think a lot of AI advances have been totally game-changing for the second question of how to make them. But a lot of times, knowing which genetic changes to make is kind of a fundamental unsolved problem. And for me, I'm thinking more about common diseases than monogenic diseases. I think often in the case of monogenic diseases, we do actually have a pretty good sense of what kind of genetic change we'd like to make. But if we're trying to kind of broaden the scope and think about how to have people live healthier lives, also for many different psychiatric or autoimmune or metabolic diseases, then trying to understand which genetic changes we would like to make is a fundamental bottleneck. So with monogenic disease, it's pretty obvious that you need to be studying human genetics. For common diseases, human genetics is still a really important tool. And I think this is very intuitive. For example, if there are carriers of loss-of-function mutations in PCSK9 who have extremely low LDL levels and are otherwise healthy, then that gives you a pretty strong hint that maybe PCSK9 would be a good target to look at. But there's also big picture empirical evidence that if you look at not just genetic drugs, but drugs overall, over the last couple of decades, that the drugs that have evidence from human genetics are more than twice as likely to succeed as drugs that didn't have evidence from human genetics to begin with. And you can see this broken down by disease area if you look at the right-hand plot. The relative success, the amount that having genetic evidence helps. So there's a lot of different kinds of genetic evidence that you might have saying this is a good target, this is something that we should think about trying to drug. The most direct type of evidence that you can get for a common disease, or one of the most direct types of evidence that you can get for a common disease, comes from what's called a rare variant association study. So in a rare variant association study, you have cases. So here, each row is a different individual. There's cases with the disease that you're studying, and controls are individuals without the disease. And every column is a different potential mutation that would give you loss of function in this gene that you're studying. If the gene has nothing to do with the disease, then you'll see the same rate of loss of function in cases as controls. On the other hand, if loss of function increases the chance that you're going to get the disease, then you're going to see more loss of function mutations in cases than controls. And so in a rare variant association study, what we do is we just ask, is the rate of loss of function in this gene higher among cases than controls? And if we can do this at statistical significance, then it gives us an extremely interpretable result, often with a pretty high effect size. And this gives us causal human biology. So before I get to the AI, let me break things down just a little bit more. So here's data for one of the genes from a rare variant association study for schizophrenia, which is a notoriously difficult disease to study. In a data set that had about 4 times as many controls as cases, there's a visualization here. So you can see the gene with the exons horizontally, and then the protein-truncating variants that cause loss of function in this gene broken down by cases and controls. And you can just look at this. You don't even need a powerful statistical test. There's no AI going on here. You have 4 times as many controls, but you have many more protein-truncating variants as cases than controls. This is not a random gene. This is a gene that has something to do with disease. And specifically, this is a gene where if you lose function in this gene, it increases your risk of disease. So where I'm going with this is missense variants. We know that protein-truncating variants lead to loss of function, but missense variants are 10 times as abundant as protein-truncating variants. So if you look at this exact same gene and you just try to visualize the missense variants in cases and controls, then here there are more missense variants than you would expect in cases versus controls, but it's not nearly as strong as a signal. either from an effect size or a statistical significance point of view. And the reason is that missense variants, they cause a change in the a single amino acid substitution. And there are some amino acid substitutions that do nothing to the protein, and there's some that cause loss of function. And then there's some that affect a very specific aspect of protein function. And so if we could figure out which missense variants do things and which missense variants don't do things and what these different missense variants do, then that would be game-changing, not only because it would greatly increase the power of these rare variant association studies instead of looking at this very sparse signal that you get from protein-truncating variants where you've got this small handful even after collecting tens of thousands of cases and controls. If we could unlock missense variants, then we have the potential to do the equivalent of doubling our sample size just from computational analysis. And moreover, understanding mechanism is a really important open problem for following up on these kinds of associations. And because missense variants can have very specific effects on protein function, understanding missense variant effects also has the potential to help us with mechanism. So this is a very well-studied problem. It's been recognized as crucial, and it has gotten a ton of attention. The Atlas of Variant Effects Alliance maintains a spreadsheet of variant effect predictors, which last time I looked at it had 117 rows, each row being a different method for characterizing variants. And these methods are getting a ton of attention because this problem is rightly recognized as super important. And the methods are so cool, right? So there's message passing algorithms on protein structures. There's large language models trained on hundreds of millions of protein sequences. We've got variational autoencoders trained on multiple sequence alignments to learn the underlying distribution. This one uses the embeddings from the large language models, but then uses a population genetics model fit to population genetics data in order to calibrate across genes. This method takes a similar calibration across genes approach, but it uses Gaussian processes and combines both the large language model embeddings and the variational autoencoder predictions. They fine-tuned AlphaFold. Illumina did this crazy thing where they've got, they take the predicted structures, they voxelize them, they do CNNs on the voxelized protein structures, and then they have 3 different loss functions, including loss functions that ask how well their model is matching other models, which are the variational autoencoder and the transformer. Anyways, people have thrown a lot at this. But it's still an unsolved problem. And there's a lot of different ways of seeing that it's an unsolved problem. One of my favorite ways is to say, can we recover what we already know exists? So there's 10 genes that we know are associated with schizophrenia in this data set that I showed you data from at the beginning. They were found with loss of function variation, but what if we had only had missense variation? Could we have found them? And this really surprised me when I saw it. So each row is one of those 10 genes, and then each column is one of a small handful of the methods that I just told you about. And it totally depends on the method. No method is winning. There's no individual method that you can choose. And trio, for example, in the 4th row, you can't find it with any method, but trio is a Mendelian disease gene. And if you look at, if you restrict your analysis to the part of the protein near the pathogenic variants, then all of a sudden the signal pops out. So the methods, none of the methods could find trio, but the signal is clearly there. So we've looked a little bit at why do different methods do well at different tasks? And to do this, we took all of these different methods, or some of these different methods, and we said for every gene, we're going to put the top 5 most predicted pathogenic variants and the bottom 5 most predicted pathogenic variants into this positive set and this negative set. And then we can ask, what are the kinds of variants that these different methods are caring about? And you can see that, and then we can characterize them biochemically. So for example, all of the methods agree that destabilizing mutations are more likely to be pathogenic or deleterious than random mutations. And they all agree that if you have a mutation in a disordered region, it's less likely to matter. But how much they care about it is really different. So green is this primate AI 3D method. It cares a lot whether something is buried or destabilizing, much more than alpha missense does, for example. What really surprised us is that they really care different amounts about the actual amino acid that's being mutated. And so if you ask, is it a good thing or a bad thing to mutate a non-polar neutral amino acid, then alpha missense thinks that's probably okay. And primate AI 3D thinks that's probably a really bad idea. So I'm going to close with just pointing out that this is more complicated than just a one-dimensional question of how likely is this to be loss of function or pathogenic. And I'm going to do that by coming back to setD1a. So setD1a has a RNA recognition motif domain on the left and a set domain at the right, which is the known active catalytic domain. It's a DNA histone methyltransferase. And it turns out that all of these different methods, they more or less agree that both the set domain and the RNA recognition motif are important, and also that this mysterious region in the middle is probably important as well. Despite the fact that the set domain has received a lot more attention and is generally more well studied than the RNA recognition motif, we developed a method to look at just clustering of disease mutations in 3D space on the protein clusters. And what we found was actually that for schizophrenia, we're finding a cluster all the way in the RNA recognition motif, which initially I thought might be like a problem or a false positive until I saw that all of these pathogenicity predictors were saying, no, that's a really important part of the protein. But in autism spectrum disorder, we're finding a totally different signal that's right here in the middle of the region. So in conclusion, we should be thinking about target identification. Human genetics is a really important lens for that, and missense variance characterizing missense variance is a crucial unsolved problem. So great. All right, thanks very much.